welcome to Bench Strength. I'm your host, Dr. David Dismuke. One of the best parts of my career has been getting to know the scientists, entrepreneurs, and visionaries. I've learned as much over the years through conversations with my peers as I have through publications and presentations. And that's what's inspired Bench Strength. I wanted to create a space where we could have the kinds of conversations, cover topics that don't fit into a 20-minute conference talk. We'll hear from scientists, engineers, clinicians, and industry leaders who are building this field, what they're working on, what they've learned, and where they think we're headed. Every great team has bench strength, and so does AAV. Whether you work in gene therapy or just simply interested in the science, I'm glad you're here. Welcome to Bench Strength. I had the pleasure of working alongside Dr. Maritza McIntyre a number of years ago at an AAV startup, and I'm very excited to have her share her unique perspective today. We've both been fortunate to have been in the industry for many years, and we share a passion and a vision for what this field could look like. Before we got into the interview, I gave Maritza a tour of Forge, where we got into the topic. Take a look. So anyway, this is Main Street, just is what we call the mural here from Miss Birdie. But obviously, with the scale that we're producing at here, I'm curious if you go back to 20 years ago, did you have this in your mind?
Absolutely not. I mean, do you remember when we were doing cell stacks?
Cell stacks, yeah. I remember doing plates.
Right. So cell stack was an innovation, right?
Hyperstacks.
And then hyperstacks. Those were those were the scales that we were operating in.
Right. And so it's a different world. It's taking it to this industrial scale,
right,
which I think is important for the field. And back from your background, from a regulatory perspective, I'm curious how you view that.
So, as a regulator back in the day, when people had 100 cell stacks, hyperstacks, whatever, and then there was a lot of pooling going on. This makes it makes a regulator's job a lot easier because they're not. I mean, that was when your head would kind of start, the sirens are going off and well, what kind of controls do I need from them? And
because you get one contamination, one plate, the whole thing's gone.
So awesome. This is, I think, the reason why we have industrialization in anything is because it makes everything easier, right?
Exactly.
More efficient, more consistent.
Yeah. Maritza, thank you so much for being here. It's been a pleasure today to have you at Forge. This is our inaugural Bench Strength podcast series. My first time hosting a podcast. I have no idea what I'm doing, but maybe just for everyone listening, can you maybe tell a bit about yourself?
Yeah, sure. And thank you for having me. It's been a great day. So I am a virologist by training, and I have been in the gene therapy development space for almost 30 years. And the relevant experience started when I was at FDA. I was there basically not at the very, very beginning, but close to the beginning. And so just really had an excellent time there. I'm trying to figure out how to regulate the products at the same time that they were being tested. So it was an exciting place to be, and I think because of that, I developed a real deep understanding of the philosophies behind FDA's regulatory approach for gene therapies, which I have then, since leaving 20 years ago, been applying as a consultant or in my time at various startups.
And so what period of time did you join the FDA? As a started cell and gene therapy there? What time was that?
Yeah, so I started at FDA in 1998. And so I was there for, you're old enough to remember the Jesse Gelsinger. Yeah, I was there at that time. Yeah. So
and your background's a virologist.
Yes.
And so maybe how did that help you when you got to the FDA?
As a virologist, as you know, I mean, most of or a lot of the modalities are using viruses because of their abilities to enter a cell and express their payloads. And so I had that kind of knowledge of the properties of different viruses, and so that's how I got the job in the first place. Was just I put in an application and just to the FDA, like generally, and got a call, from the Division of Cell and Gene Therapy saying, "You're a virologist, come to an interview." So, yeah.
Very, very good. So, so this series that we're doing here is called Bench Strength, and so there'll be some sports puns that we'll sprinkle in here and there.
Is that a sports thing?
Yeah. So I'm gonna start you out with a one question and sports question. So in baseball, it's whenever your batter's coming up to bat, he'll have a walk-up song.
Right.
If you had to choose a walk-up song, the rah-rah walk-up song, what would that be for you?
Oh, 1999 by Prince.
That's a great one, actually. That's awesome. Yeah. Of course, I played that a lot in 1999. Yeah, but yeah, that's a great. Prince is a phenomenal artist. So, no, that's a good one. So maybe moving, you kind of talked about your bench strength, kind of where you sit on the bench. From the past, can you maybe talk about what you're doing now?
So I have over the years, I like to take what I've learned doing one thing and apply it to the next thing. And so at this point, having gone from FDA and my particular appeal and value add to either clients or the startups I was in was really the knowledge of the regulatory space. But then especially like within startups, getting pulled into fundraising, discussions, it helped me think about, like what's important, what are investors looking for, and so just combining all of that knowledge and into advising clients, and that's why I like to work with early stage clients. I most of the time as a consultant, if you are working with someone who's in late stage, they're in trouble.
Okay.
And it's a lot more rewarding to me to help people build things and build them the right way from
Makes
the
sense
ground.
from the start. Yeah. So from that viewpoint, do you have like a framework or a rule of thumb that you sort of apply across different clients as they start out? There's a lot of different products, I'm assuming. And
yeah, there's a lot of different products, but in general, there are some rules of thumb in terms of like you have to start by looking at their science. Like what is it that they're looking to do? What data do they have? And then to start helping them lay the foundation. Like so, maybe with a lot of gene therapies. That there are elements in their constructs that are just not going to pan out. And so to help them understand in sequence, like, well, you might want to change that before you get to dose range finding, for example, and just building, a logical development path.
right. And I guess you've seen some examples where clients maybe had poor design. They had to kind of just take a step back and sort of rework their idea a bit to kind of take a step back to go forward.
Yeah. Yes. And I think one of the things that I've found most frustrating is when they've gone so far that they don't have the ability to go back. And then everything we're doing from that point on is managing around a poor design.
Yeah, they're like, that'll be Gen 2. Is
Right, yeah.
Yeah,
Exactly.
So let's move forward with this when we're too far down the line. So trying to interact with them early and find those fixes early is important.
Yeah.
And is there, I guess, how do you, for that sort of perspective, how do you find them? How do you find the fixes? Or what are you looking for? Like analytical, or preclinical work? What kind of leads you say, hey, we need to stop here?
So I'm trying to drive people so that it is analytical when they find it, which means that they're doing that means that they have to perform more analytics than what someone would typically do before they put their vector into, a disease model for the very first time.
Right. And I guess it's a sort of a changing landscape out there. There's
Yes.
companies now that kind of focus on this. Do you see some of that where clients are going in and doing, before they get to their lead candidate, for example, that they do more on manufacturability or product quality? Yeah.
I am seeing that now, and it's been a long time coming.
Yeah.
And I have to compliment, CDMOs like you who offer that, because I think that one of the tensions for many years was the providers of the analytics were not playing in that space of small scale.
Sure.
And so I had this idea of, people should be looking at those things as part of candidate selection, not just, efficacy readouts in their model.
Right. I think it can be misleading because you can have a poor vector sometimes that you put enough dose into a mouse, it will work. It'll repair itself.
Yeah.
You'll get some cross packaging trans splicing that'll occur. I'm curious. What we do here a little bit is be able to apply some of these newer tools. NGS, mass spec. Better analytics that can answer those questions, especially with either smaller amounts of vector or you're getting higher productivity, so you can run like other assays. I'm curious if you see some of that. Yeah.
Well, I think the analytics that require smaller samples have been a huge boost to the field and to applying that kind of a principle of quality by design at the candidate selection stage.
Right,
yeah.
Exactly. So I'm happy that we're doing that here, but I'm also happy to see other companies sort of stepping in and utilizing even AI tools and things to help parse through the data and find those better candidates. I think it leads to lower doses, more effective drugs. So,
Right.
Let me ask you sort of, again, back to the sort of sports puns, looking at your career and thinking about, again, you're talking about having, stepping in and sort of giving this advice to your clients in companies you've been in. Have you had sort of this clutch moment where you can sort of save the play, as it were? You have example of that you can maybe share with everybody?
So I was working with a client, a company, and they were, the agency was having, they expressed concerns because there had been HSA filled on this same line or I think it was HSA. And then I just, because I knew, the products that FDA was regulating, and I kind of launched in and I said, come on, you've got cell therapies going into people and you're not, wringing your hands over HSA there.
Sure.
And that kind of helped shift their frame of mind. Yeah.
It seems like it's an interesting question, because cell therapies obviously is more complex even than gene therapies. You can't apply some of these, you can't do sterile filtration, you can't do that. So you have to rely on other analytics. And so do you think that gene therapies are treated more like cell therapies or more like general biologics?
I think more like general biologics. Yeah.
So the complexity is not always fully considered.
Yeah.
Okay.
Yeah.
I think that's true from my experience as well. Back to more of the sort of the technical side of things. We're thinking about how the design goes into maybe helping catch things earlier and better analytics. Do you think there's other ways that because it's always a bit of speed to the clinic and trying to get therapies to patients as quickly as possible. On the preclinical side, do you think that we're doing anything there with cell-based models and things like that?
I think that's a space that's really growing, and the agency is becoming a lot more open to it. And I think that'll help with development of a lot of gene therapies where there's now a model. Sometimes you can knock out a gene, and in a mouse it's just not going to have phenotype, or it'll be variable. Like one litter they will be affected, and the next one isn't. So it becomes difficult to near impossible to do your drug development in those models. So.
Yeah, and the whole idea that mice lie, and I forget the one about monkeys, but it's
mice lie and monkeys don't always tell the truth.
Yeah. So trying to find models, especially avoiding animal models, is a push from the agency, which everybody thinks is aligned to, but then trying to find these good in vitro models that can be predictive. And one of the things earlier today we were talking about was, sort of that with. It's always the push to find, get the clients signed up for the trial. And do you see pharmacogenetics playing a part of that? Understanding sort of the gene background of patients and how we're selecting patients for trials, and understanding who's probably the best candidate for a trial, or who's the best candidate for a therapy.
Right. I think that might be going on more in the more small molecule space. That would be. I haven't seen it that much for gene therapies.
It seems like it should be applied. Obviously we're going for there's different alleles that you're going after potentially, and maybe there's in other, especially from epigenetic. Maybe I don't know, but it seems like there's probably a way to apply more pharmacogenetics to how patient selection's very critical, and why is there variability in the patients that we're choosing for our trials,
right?
And how do we explain it?
Right.
I don't know how else to explain it, but yeah. So
it would be interesting to say that to a clin dev person, because they're always stressing about being able to recruit, and that's probably the last thing they want to hear.
Look at. That's right. trying to find the right. Yeah, I mean, we do see a shift, at Forge for more larger patient populations, more chronic diseases. We saw those earlier, with other diseases going after our heart. Some of them have some resurgence. And some of them, in my opinion, were, failures because, again, back to animal models in sheep or something else that would show the clinical benefit at a certain dose, but maybe you don't see that in the patients. Have you seen more of a resurgence for some of these larger patient populations?
I think that for cardiac, there's definitely a resurgence, and it's pretty exciting, I think, because it seems to me like the heart is a lot more amenable. You can actually really correct things.
Right.
So that's exciting.
Yeah, I agree, and I think we're getting better vectors back to design, of the transgene. I think we're kind of focusing there, but I'm excited about sort of this convergence of being able to apply these tools for better transgene design. At the same time, so many different companies have been working on novel capsids.
Yes.
And that novel capsid, and then better transgene design, and then better manufacturing, better analytics. Hopefully, I think that's really driving us towards better effective drugs.
I think so too.
Yeah. So I don't know if you kind of work with some of those clients, or it's still a lot of the rare diseases or that sort of dominate.
I have a little bit of both. Yeah. And so it's still most the dominant is rare diseases, but I have clients who are going after larger diseases, and a lot of times then the questions from the VCs in diligence are can you really make enough vector?
Yeah. We met with a client not too long ago that's going after a disease model, and they looked at it. It was local administration still, but it was going to basically require every CDMO that makes AAV to make their product. So, so that's it's a challenge still, I think. So.
Well, yeah, especially when you think about using AAV for vaccination.
Yeah.
That is when you really start getting. Can we actually supply that? Yeah.
We did get during COVID, which is an interesting time. I kind of feel like we should have taken them up on it because we were struggling at that point in time to get materials from everybody. COVID got it first, manufacturing. So we did get approached to make COVID AAV. So, I think that's a strategy that absolutely could work. Yeah. Obviously, adenovirus has worked. It uses very well as a vaccine. So. We'll see where it goes. I'm going to have to continue to follow that one. So right now we have a lot going on in gene therapy. Is there something in particular that you would look at and say, this innovation, this thing that we're working on right now really changes the field. This hinges, what we've learned in the last, say 5-10 years that is so important that we've maybe missed earlier.
That's solved, or that is a potential solution?
That's maybe so it was a problem, maybe, and then, or an innovation that you feel like has really changed the field.
So it's already changed it.
I guess it would say it's already changed.
Already.
We're changing.
Well, I mean, AAV,
okay.
right?
Sure, yeah. But within AAV specifically.
But within AAV, I think that just like the work that went into identifying all the different serotypes. We started with AAV2, and it, for a variety of reasons, was not, the best.
Sure.
And so that to have this menu of different, first of all, just the natural ones, and then the ability to engineer them. I think that has been game changing for the field. We had AAV2. We wouldn't have a Elevidys, right? Like you couldn't have.
You jump there. Yeah.
Yeah.
I'm curious, for, obviously we're trying to make strides here in making more more vector per batch, more doses per batch. And so really trying to push up that productivity. But we don't, we're not really in the clinical side. On the clinical side, I'm curious what you're seeing from the immune side, and how is that landscape changing? It
is, and it needs to change. Okay. I think that we understand now that this is a problem, particularly with high dose, and so we either need to lower the doses by somehow either engineering the capsids to escape, improving the payload so there's less, innate immunity. But something that I'm really excited about is the ability to just clear out those pre-existing antibodies for a short amount of time.
Yep.
Yeah.
And for this would be weeks or just during the administration phase.
Yeah. So there's now I'm blanking on the names. There's the
IDZ. Those molecules.
And some of the gene therapy companies have licensed that and are exploring it. And then there's, others.
And you think that actually opens up the door to redosing? Or is it really about that first treatment?
Potentially both.
Okay.
So the way I think I would see it is first to explore the ability to both expand the pool, but also I do believe that there's limitations to our understanding of when we're screening. For most gene therapies, they pre-screen for pre-existing antibodies, and the question is is it sensitive enough? And it appears that it might not be, and especially there's been a switch from looking only at neutralizing titers to total, because there are a lot of other antibodies within a pool that they're not going to score on your neutralizing titer, but they could have impacts on both actual biodistribution and transduction efficiency, but also safety. Right. So if you could just take that out of the equation by clearing them all out for, a week or so.
Right. And so then, obviously a little bit more patient care, but at the same time for these especially critically ill patients to be able to, who are not candidates because they have neutralizing antibodies or antibody titers are too high.
Antibody titers, pre-existing immunity. Yeah. I think that I would like to see that really tested more.
I'm excited about it too. I do, especially from a biodistribution perspective, or there's always, continued innovation, and the capsids may be too similar, so you can't redose.
Right.
I
don't think that just switching capsid is going to do it. So to be able to redose, you're going to need to have something more effective, like clearing them out. Yeah.
Agreed. Kind of leads me to a different question. So again, back to those sports puns. So for training camp, is what we're calling this one. Is there anything that you see from your background, and all that you seen? Is there anything that we feel like for people who are in the field, especially in startups, in companies, that really they need some more training, like the training that would be helpful for them to understand as they develop their drug?
So something that I wish, and not everyone has to end up being well versed in every single regulation in the code of CFR or guidances, but there's, I think, the framework that FDA operates under. If people could understand that concept, because it actually becomes really simple. That you can't, the authority comes through commerce, and that you're not allowed to sell anything or put it into interstate commerce that is mislabeled. And so if you say something is going to cure something, but it can't, then it's mislabeled or adulterated. And so then, if it's contaminated or it doesn't have the strength, et cetera. And I think that if people could understand those, what the ultimate goal is, what FDA is looking for,
Right.
It might help
When they're designing.
Because then you think about it as you're designing your trial, as you're designing your product, your analytical, package, et cetera.
Right. Yeah, I see it a lot of times. Obviously, in the device world, it's a little bit. It starts with design, right?
Right.
And
exactly.
so I think sometimes that is sort of skipped over in biologics. It's like straight to I've got a candidate, let's move forward. But all the design wasn't all fully considered. So I think that's important. So that's where I see the future is pretty bright, because we have all these a lot better tools, as far as training camp and thinking about how we train individuals. Just so much access right now. Obviously, you can jump online and learn anything, right? And so it's a lot more accessible. And I think COVID even helped it, now for people to get even additional degrees, to continue to take courses online. So I think the future is very bright. Is there one thing in particular that you're super excited about as you look ahead, for gene therapy, cell therapy, AV, anything in particular that you think is super exciting?
We've learned how to make them a little safer. We've also learned we are learning and starting really to pick the right tool for the right target. And so I'm pretty excited about just having more robust efficacy data coming sooner.
From a preclinical efficacy?
No, in the clinic.
In the clinic,
I think that I feel like we're on the cusp of really having de-risked a lot. And so once the trials start, we'll have some pretty clear signals quickly.
Yeah. One thing that was surprising to me when I got into AAV gene therapies, you hear about phase 1 trials about safety, and one thing people don't know about me is I was probably a part of about 25 clinical trials in grad school, so I made some money. So, but I was a part of all those clinical trials. It was just, determining safety of a small molecule generally, and before they would move to any sort of dose escalation finding. And maybe that explains my, what's going on with me. But but for AAV and gene therapy, cell therapies, efficacy is really, although we don't say it, it is really efficacy is looked at at phase 1. And do you think that but your perspective is that really that's going to continue, and we're going to look deeper at having better signals for efficacy for these trials?
I think it will always it will continue because for gene therapies and also for cell therapies in a certain sense, even the risk profile is such that you wouldn't put it in, a college student who's trying to make his beer money. But then also you just because especially for gene therapies where it's especially for genetic disorders, like you're going in to fix something. And so I don't think it will ever change that you're starting off in those populations. And if your gene therapy is designed to stick around forever, why that would be a waste of, a valuable patient resource to not look for signs of bioactivity at a minimum, right?
And do you think it changed? Because obviously typical pattern is these dose escalation trials. Do you think that the body of knowledge that we're learning, helps to maybe tighten those windows of what you would do in a dose escalation, or maybe avoid some of it in a way?
I think so, and especially like if you look in ocular gene therapy, there's not a lot of dose escalation because there's a pretty tight range, right? I mean, it's a small space, but also there's a pretty good understanding of the dose, the dose window within which you can, get good correction. Right. Yeah.
Makes sense. So I shared a little bit about me of something people might not know. I'm curious if there's anything about you that people may not know. It would be an interesting fact.
So my daughter, she showed horses throughout her childhood, which now I pay for 2 horses to eat grass in North Carolina. But when she went away to college, then I started learning how to ride.
Okay.
Yeah.
Is there a first time you hadn't ridden horses before?
No.
Okay. Yeah, interesting.
Yeah. So that was fun.
Okay. And are they like big horses?
They are. Yeah. And actually one of them I can't, before they're retired now, but one of them I couldn't ride him because he was too powerful. It would be driving a Maserati. Like once you're on his back, he just starts to go. Okay. Yeah. So.
But your daughter was.
Oh yeah, I mean she did show jumping with him, so you have to go do a course and jump over these jumps in the right order.
So if you were getting, he's like wanting to jump over the fence.
Yeah.
Okay. No, thank you. Yeah. Well, that's all the questions I have. And maybe is there anything else that you kind of want to share or anecdotally
I guess I just want to say that I'm really excited to be here and to see what you guys have built here. It's very impressive. And as I was saying before, soon after I left FDA, suddenly I found myself getting taken on all of these tours of facilities, and it was just very odd. And because CMC reviewers are not, the people who from FDA who go through your facility, that's a whole different group.
Sure.
Right. And they know everything you need to know about your water filtration system. And that isn't something I knew. And I would chuckle to myself because that actually would always be the big reveal. And now we're going to take you to show our water system. Right. And it's just I really don't want to see. Or the warehouse, it's a big deal to show a warehouse.
Yeah. And so, we're very proud of our warehouse.
Yes.
and our air handlers, we're very, very proud.
I'm glad I didn't have to pretend that I liked her water system. But,
okay.
Yeah. But it's nice. It's a little, I don't know, full circle, but it's nice. I haven't done it in a long time, and I enjoyed it. And I especially enjoy it because of how much you guys are contributing to advancing the field.
Thank you. So. Well, again, great to have you here, and come back often. So we love to have guests in Columbus, so we'd love to have you back as much as possible. Thank you so much.
Okay, thank you.
Bench Strength is produced by Forge Biologics. All rights reserved. No portion of this podcast may be reproduced, distributed, or used without prior written permission from Forge Biologics. Learn more at forgebiologics.com.